Okay, I guess we're ready to get started again for this next session. Just briefly introduce myself. I'm Jonathan Allen. I'm a bioinformaticist at Lawrence Livermore National Laboratory. My background is primarily in computational genomics, and I'm sort of a genomic generalist working on a wide variety of analysis problems ranging from microbial metagenomics to human SNP characterization. So it's kind of a wide span for me. But I'm very interested in all of the sort of the tool development questions and the challenges that we face in terms of developing measurements and to assess what it is we're seeing at the microbial community level. So it's, I don't want to take too much more time in terms of introducing myself. I want to move on now to this next section of the panel. And sort of the theme of this next session is on understanding the current toolkits that are being used to study microbiomes in the built environment. And just because this is such a complex set of observations that we're trying to make in terms of microbial communities, this is really sort of, I think, an important topic that we need to wrap our heads around. And it's my great pleasure to be able to introduce Rob Knight, who's really been a pioneer in terms of developing tools in this area in terms of studying microbiomes in a wide variety of environments. And he's a professor of pediatrics and computer science and engineering at UC San Diego, and he'll be talking about sharing his perspective on this topic. So without further ado.
Okay, thanks, Jonathan, and I'd like to thank the National Academies and especially the Sloan Foundation, who've really been key in funding this area, for organising this meeting and for inviting me to tell you about some of this stuff. So I'm going to explain some of the methods that you've seen used in some of the earlier talks and some of the ones that you'll see later on. And just to reinforce the point about why it's relevant to see these talks about permafrost and soil in this context, it's important to remember that we do live in a microbial world. These complex microbial biofilms appear anywhere. This one's out of the ocean, but you can see an equally good one out of sputum or out of soil. And the amazing fact is that there's 100 million times as many bacteria on Earth as there are stars in the universe. So each teaspoon of your stool contains the amount of data in the genomic DNA that it would take literally a ton of DVDs to hold. So what this means is that in many ways, microbiology is the ultimate in big data science. And this has been driven by the decrease in cost in DNA sequencing, which has got 10,000 times cheaper in the last 10 years, about 1 million times cheaper in the last 15 years. Because you wouldn't pay $100 million to find out what's in your gut, unless you're Donald Trump or something, I guess. But you might pay $100 to find out what's in there. And one thing that's important in this context is that we used to use microbiome mainly for discovery science, and that was the enterprise that those of us like Janet and myself who were doing this 10 years ago really introduced these tools for. And today's tools still reflect this to a large extent. So this is an example of a paper that was state-of-the-art in 2005. So I don't want to pick on the authors too much, but this is something that was published in Science, looking at new microbes out of the ocean. So you see this phylogenetic tree with a handful of new sequences in bold here that are plugged into that phylogenetic tree of existing knowledge. And then they did some clustering of the 4 samples that they looked at to relate those 4 environments to one another. And so what you're seeing represented today is progress in just 11 years over this kind of study of a handful of sequences and a handful of environments. So with this rapidly accumulating data, we took a leaf from a very old book to try to get a handle on it. And so specifically, for those of you who haven't read Darwin's work on the origin of species recently, or perhaps at all, the first edition is often considered a pretty dense read. So it has 502 pages and only 1 figure. So people who read it for the pictures are often disappointed. But I'd argue that you shouldn't be, because the one picture he included was this idea of the phylogenetic tree as a way to get a handle on biological complexity with modern organisms, which we now represent as modern DNA sequences out in the tips, and then hypothetical common ancestors that group them together that let you infer shared properties across groups. And so Kathy Lozapone, a very talented PhD student in my lab at the time, introduced this concept of unifrac to exploit evolution to compare microbial communities from anywhere, whether they're in the oceans or the soils or our homes or our own bodies. And the intuition here is very simple. The idea is that if you have two unrelated communities, so remember the tips of the tree represent modern sequences, the different colors now represent environments. And the idea is if you have two distinct environments, nothing that lives in the red can move into the blue and survive and reproduce and accumulate the molecular change that leads to evolutionary branch length on the tree. So all of the branches lead only to members of the red or to the members of the blue community, so they're all unique, colored in red or blue. None of them are shared by both, and we define that distance as 1. Whereas in contrast, if you have two identical communities, everything you see in the red, you see in the blue. All of the branch length is shared, shown here in purple. None is unique. We define that distance as 0. And then for any two actual communities, we'll have some unique branches in red or blue, some shared branches in purple, and some distance between 0 and 1. And so what's great about this is we can exploit Darwin's insight that there is one tree of life that unites bacterial communities and microbial communities, and indeed all organisms from anywhere. And so we can take 3 communities, red, yellow, and blue, paint them on that one tree, and compare red to yellow, red to blue, yellow to blue, taking into account all the evolutionary history of the tree. And we can store those distances in a distance matrix, and then use techniques like principal coordinates analysis, another one you heard about is NMDS, to reduce the information in this matrix into a map of where the microbes are, or you could do hierarchical clustering or all kinds of other multivariate analyses developed over the last 60 years. So what's so great about this, you're probably thinking? Well, Kathy, with this technique in mind, Kathy initially decided she would look at everything. And so she assembled from GenBank a data set of over 20,000 sequences, which at the time was a massive number of sequences spanning over 110 different studies. But today it's probably your rounding error per sample in the data you get back from the sequencer. But anyway, this set of samples literally spanned the globe, so everything from the poles to the equator, extremes of temperature, of pH, just about any biotic or abiotic factor you could imagine. And the question was, out of all this complexity, could we see patterns linking all those microbial communities? And amazingly, the answer was yes. So we saw this very clear split between saline and non-saline communities. You see a few communities in blue in the middle. Those are things like estuaries, where you truly have a mixture between saline and non-saline. So out of all this microbial complexity comes a very simple pattern. And salinity was a long way down the list of factors we thought would be important. So if we look at hot environments, whether it's from a hydrothermal vent at the bottom of the ocean or a hot spring in Yellowstone, those just cluster with the other saline and non-saline communities respectively. So temperature is not the main driving factor, nor the other things on that list I showed you. However, in subsequent work with Jeff Gordon's group, we were able to show that there were some really extreme microbial communities out there. And when we look at these microbiomes, they squash all of these environmental communities from the saline and the non-saline communities all over the planet into just one side of the graph. They introduce this new axis that explains twice as much of the variation. And you might be wondering, well, how far do you have to go to find these microbial extreme environments? To the top of a mountain, to the bottom of the ocean, or what? And the amazing fact is you don't have to go far because they're right here within us. So what we're looking at here is the vertebrate gut. In between, we see things like the mouth, the vagina, and so forth. And so what we see is this fundamental distinction between these biotic and abiotic communities. And we can even do this in a network context. So what's amazing about this is that we see particular kinds of bacteria, especially, so you see this set of free-living bacteria, and then a set of human gut and vertebrate gut. Then in between, we see things like the skin that provide an interface between the host-associated and the non-host-associated environment. And as you've heard, a lot of the composition of the built environment comes from the humans and other animals that contain it. So we didn't have that data at all back in 2008 when we put this figure together. But since then, so much information has accumulated about the built environment, truly revealing its role as another interface between these fundamental splits between host-associated and non-host-associated microbes. So as I mentioned, a lot of this was pure discovery driven, but now we're starting to ask questions that really matter to people, whether from a health perspective or from a regulatory perspective, and that's where we really need to start thinking about new tools. So for example, my lab had a collaboration with the FDA looking at the load of salmonella on different parts of the tomato plant. And they got very excited about the data on the grounds that they were seeing very differentially abundant loads of this pathogen depending on where the crop was on the field and where on the plant it was. And they were all excited to publish it, but when I looked at the data, another organism that was just as common as salmonella in their samples was the duck-billed platypus. And this has also been seen in various other contexts, like the built environment context as well. And so the issue here is that it's very easy to misidentify things. And when we go to MGRAST, a very popular resource online, and we ask about the distribution of organisms, just where are they in the world most relatively abundant, the organism that I'm highlighting here, that is most common in Finland apparently, is indeed our friend the platypus. And the reason for this is that rates of misassignment are very high, and that's what leads to reports of things like anthrax and plague thousands of miles away from where those organisms truly live in the environment. And so extending this work, we were also curious about extinct species like the dodo and the thylacine and so on. And the amazing fact is, if you take these results at face value, a lot of these charismatic animals that we thought were extinct, you can find in fecal samples all over the planet. So either there's dodo farms and thylacine farms and so forth, where they're being kept in secret and people are eating them all the time, or we need to substantially rethink the methods that we're using to make these assignments. And if you think about what's happening in these situations where it's just obvious what's going on, imagine how terrified you should be by all the results in the microbial world where all of this is invisible. So in the Human Microbiome Project, one goal of the NIH with this large-scale project, so $173 million, and my lab was involved in this in several respects, but we're talking about a large community of over 400 researchers, and Owen White, who's here in the audience, was the leader of the Data Analysis and Coordination Center that put all of this stuff together. What we were doing here was trying to put together a reference baseline for what does a human microbiome look like. And so one of the that was one of the key components of the project. There were also demonstration projects linking the microbiome to disease, technology development projects, and so on. But one of the key components published in Nature in 2012 was looking at up to 18 sites on the body of up to 250 people. And Janet already explained the technologies, so I dropped that part from the talk. But we did a combination of 16S sequencing to get a microbial taxonomic inventory and shotgun metagenomics to get a functional inventory. And this was 4.5 trillion bases, so 1,500 times the size of the human genome sequence in this one project. And so the drawback to it is that we did get all these DNA sequences. So here I'm just showing you the first file of data from the HMP. So fundamentally this is an ecology exercise, but it's pretty hard to tell who lives where in the environment from this kind of data, right? Like you probably can't even tell that that's an oral sample, let alone the sequences that would let us make that call. And you might be thinking, well, I need to see the taxa and the functions to figure it out. But when I show you that, it's not really much more informative. So each line on this is a stacked bar chart of the microbiome of one individual, that individual's functional repertoire down below. And what you can see is that even if we just look at the stool, there's tremendous variability one person to the next. So some people are mostly Bacteroidetes, others are mostly Firmicutes, others have a lot of Proteobacteria and so forth. And what's amazing to consider in this context is that you're 99.9% identical in terms of your human genome to the person you're sitting next to, but you might be 90% different in terms of your microbiomes. And so all of this diversity is really a problem because the technical capacity to read out the microbiome now exists. So in crowdsourced projects like American Gut that I'll tell you about later, and there's various companies that are doing this kind of thing now as well, patients are coming to their physicians with their microbiome profile and saying, hey, I got this readout of 1,000 species that were in my gut. Or even better, I got this readout of 1 million genes that are in my gut. So can you tell me what's wrong with me, doc? And you can just imagine what your physician's reaction to this is, right? All they can do is refer you to their colleagues in psychiatry for being crazy enough to think that that's going to help, especially in those 15 delightful minutes that you have together at best, right? So the challenge for the field is to make it not crazy anymore and to figure out how we can take a single profile from your body or from your home or from your office and integrate it with other information so that you can actually get something out of the data rather than looking at the profile in isolation. Now, to facilitate this, the main thing that's important is being able to move the methods to higher throughput. And this is something my lab's been working on for about the last decade. So in 2008, we introduced this technique of highly multiplexed barcoded sequencing using formal error-corrected codes that we introduced into the PCR step. We mix the samples together and sequence them on 454, although more recently we moved to the Illumina platform, which is more accurate and higher throughput and cheaper. And then we use the barcodes to assign the sequences to the samples, trim the barcodes, group them into what are called operational taxonomic units, so basically proxies for species, and then use the techniques I showed you before to make these maps of where all the samples fall. And the software that does this is called Chyme. It stands for Quantitative Insights into Microbial Ecology. It's free, it's open source, it runs everywhere from your laptop to the Amazon EC2 cloud to the Hopper supercomputer at NERSC with 120,000 cores, which is the largest system we've run it on so far. And it's really been a community effort to develop this software. Greg Caparasso really led that development when he was in my lab, and he's now an independent faculty member at NAU. So anyway, with this pipeline in place, you can go straight from data off the sequencer to ecological insights. And although it's a common misperception that this only works for 16S data, you can already use it for any amplicon and for shotgun metagenomics, and if you do some pre-processing with shotgun metabolomics as well. So when we apply it to the HMP data, we get this much more interpretable map. And you're probably thinking, well, wait, I don't understand that any better than I understood all those sequences you flashed a few slides ago. But just bear with me for a moment. So each point on this map, and this is a principal coordinates reduction of unifrac distances, although you could have used NMDS or other scaling techniques, and you could have used other distance metrics to measure the distances between communities. But what we're doing is we're taking all the complexity of each microbial community, reading it out by its DNA, and projecting it down to just one point on that map. So 2 points that are closer together are more similar in terms of the evolution of the microbes that are in there, and 2 that are further apart are more dissimilar in terms of the evolutionary history of the microbes they contain. So if I color this by body site, you can see immediately the main thing going on here, which is that different parts of the body, the mouth, the skin, the vagina, the fecal communities, all have very different microbiomes. And so this is much more important than, say, whether the subject's male or female, how old they are, what time of day it was collected, all kinds of other things you might imagine would affect the result. And if I highlight the first person in the HMP, you see their mouth and their gut are in totally different locations from one another on this map. And it wasn't until we did the Earth Microbiome project, which Janet Jansen and Jack Gilbert and I co-founded back in 2010, that we understood how profound these differences were. Because what we could do is we could go out on the planet and ask what 2 samples are as far apart from one another as the mouth and the gut of the same individual. And if you think of your mouth as being kind of like a coral reef with complex mineralized structures covered with biofilms that your dentist bugs you about, then the amazing fact is that your mouth is as far away from your gut in terms of its microbial ecology as the water in this reef is from the soil in this prairie. And that's amazing, right? Because it means a few feet along the length of your body makes as much difference to your microbes as thousands of miles across the planet's surface. So to extend this a step further, as a collaboration between the Earth Microbiome Project and the Human Food Project, we started American Gut back in 2012. The idea was to use crowdsourcing and crowdfunding to let anyone put themselves on this map. And I'll show you the most impressive example of why you care about this, which is fecal transplant for C. diff. And so this is work we did with Alex Kurutz and Mike Sadowski at the University of Minnesota. C. diff kills 14,000 people a year. And if I show you gut microbiomes from people with C. diff, they're in this totally different part of the map. But when we do an intervention, in this case, fecal transplant from this one donor, so 4 of them are going to get transplants from this one donor who you see is in the healthy region. The question is what happens then? Do they progress towards the donor state, and is it fast or slow? Is it a subtle change or is it dramatic? And each frame in this is 1 day in the life of the microbiomes of these individuals suffering from C. diff. And what you can see is that immediately all 4 of them move into the healthy microbial ecology state, and then they stay there through the months of follow-up. And this is showing you that just as you saw this morning in soils, as we heard in the questions, we must do for the built environment, in the human microbiome as well, these intervention studies are critical for understanding the types of changes that can happen. And in this case, figuring out how to identify an aberrant microbiome state in patients and bring it back into the healthy state, whether in this case by direct transplant, but you could also imagine doing it by chemical means or doing it by microbiological means with probiotics or all kinds of other interventions, many of which have direct analogues in the built environment. And so what we really need to do here is to take, in the human case, is to take all the diseases linked to the microbiome and identify the good and the bad places in the map and how to get from one to the other. But you could imagine doing exactly the same for sick homes and sick workplaces. So as I've shown you, a lot of the stuff you can do at high taxonomic levels, but some questions are going to require us to move all the way down into the strain level. So for example, with Bacillus cereus versus Bacillus anthracis, you can't figure out the difference between those with the 16S, right? They're identical in the 16S, but one of them will kill you, whereas the other is completely harmless. And there's definitely a whole range of clinical questions where we care a lot about this. So in the Genomic Standards Consortium, and Lynn Schrimmel, who's been leading a lot of these efforts, is also here at this workshop. What we've been doing is putting together a set of standards for annotating genomes, including standards for annotating environmental samples and annotating complete genomes. And most recently, there's a paper in progress on annotating environmental genomes from single-cell sequencing or from shotgun metagenomics. And so this paper basically describes a lot of standards that you can use to annotate your samples in a systematic way and get them into INSDC, and we're using these throughout American Gut and throughout the Earth Microbiome Project. So there's a data set of tens of thousands of samples that you can directly compare your samples to if you integrate into these standards. And they're designed in a very modular way where you have checklists depending on the type of sequences you collected, whether it's genomes, whether it's metagenomes, whether it's marker gene sequences, and then different checklists for different kinds of built, so there's shared descriptors and then there's environmental packages that apply across all of these types of data. And so Lynn led this effort on MixBE, which is a specific extension of these standards for sequence data from the built environment, which was published in the ISME journal a couple of years ago, and provides an example of how we can integrate this data with other data sets, including the sources of these microbes like people and soil. I also want to highlight the microbiome quality control project. So far this is limited for stool, but essentially it's a cross-lab validation effort of the kind that you heard called for earlier, basically taking the same samples, sending them to a dozen different labs, and asking how similar are the samples across labs. And the paper describing this is in review at Genome Biology right at the moment. So in the last 5 minutes, I'm going to get into a little about how we can map microbiomes in an explicitly spatial way, which is really critical for understanding a lot of these built environment data sets. And a lot of this work started when I was at Boulder, so this is a collaboration with Noah Ferrer, where we were looking at sources of microbes in residential kitchens. So you can do untargeted 16S sequencing and ask at the genus level where's the Campylobacter, the Salmonella, and so on. But we can also use source tracker, so a Bayesian technique for asking what are the sources of all of these different communities, and ask where does the skin go, where it's mostly on things that you touch, like handles and the trash can and the floor where you walk with bare feet. Where does the produce signal go, so that's mostly on the counter, and where does the tap water go, so mostly around the tap. We also did this in the bathroom down the hall from my lab, so you can see the skin, again, mostly on things you touch, like the door handle and the toilet seat. You see the stool, fortunately, mostly in the bathroom stall itself. And then you see the soil community, mostly on the floor, but you also see some of it on the flush handle. And coming from New Zealand, I had never heard of this practice, but apparently in the US, a lot of people use their feet to flush. And so we picked that up directly from the soil traces left behind on the flush handle, which I never would have guessed had I not seen the data. And with Peter Dorrestein, who'll be talking shortly, we've also been looking in more detail at that interface between the skin and the built environment. So essentially the study design here was to take 500 samples from 2 individual people, and then Peter ran it through 2 types of mass spec, and I ran it through 16S sequencing to look at the bacteria. And what we can do is we can then map that data onto a 3D model of the body. So here blue means that there's nothing, the hottest colors like red means there's a lot of something. So you can see some molecules, just the mass peaks are represented here all over the body. some in the armpits or the groin or the toenails. And we can do the same for the bacteria, where things like Staphylococcus is in the moist areas like the feet and the nose like you'd expect, Propionibacterium in the head and shoulders, Corynebacterium more broadly distributed on the body. And so what we can do with this is we can use the spatial colocalization to guess which microbes are doing which reactions. So you see Propionibacterium on the head, oleic acid on the head and the hands, and then the oleic acid only oxidizes on the head where the Propionibacterium is. Then we can test that in the lab by incubating them together and showing that you only get the breakdown products where the microbe and the precursor are present together. And so one thing we're doing at the moment is developing this into a high-throughput pipeline to figure out what microbes are catalyzing which reactions in the environment. And we can also apply this in the built environment directly. So this is Peter's office with 4 of us sitting around the table. Here's me, here's Peter, here's Larry Smar, our director of CalIT2. And you can see that this molecule lights 2 of us up very brightly. And the molecule that you're looking at is caffeine. Peter doesn't drink any coffee or tea, whereas Larry and I drink a lot of it. And you can also see that someone spilt some on his floor on some previous occasion. But while this is undoubtedly the most expensive way to find out about that particular coffee spill, you can imagine how useful this technique could be in environments ranging from NICUs to industrial settings to even the International Space Station. So with this ability to acquire high-throughput data, this is where things like the Earth Microbiome Project really come in. And so besides collecting tens of thousands of samples and sequencing them and releasing all the data free to the community with consistent metadata, and here's what the data look like, by the way, we can also apply this in all kinds of different ways. I'm going to skip this section in the interest of time on how we're applying it to reptiles and amphibians, which are tremendously affected by their built environment. And in fact, you can basically see that amphibians require an ongoing input of microbes from their environment to resist chytrid fungal infections. So this is very elegant work that Val McKenzie is doing at Boulder that we're collaborating on. But I want to focus on the modeling aspect just in the last minute. And this is stuff that Jack Gilbert and Peter Lawson are primarily leading at Argonne, where the idea is that from environmental parameters that we can measure, we want to build these networks that predict the abundance of all the taxa. And so what's great about this is when you apply it to something like the Western English Channel, from a couple of dozen ground truth surface measurements, you can use satellite data to predict for thousands of pixels and over dozens of time points what are the microbial communities going to look like with 90 to 99% correlation. And so the idea is that you can take satellite image data, predict the microbial communities, use those to predict the metabolic turnover, and then figure out the feedback terms. And as I mentioned, we've been doing this with the UNESCO Surface Ocean CO2 Measurements Atlas. We've also been doing the same thing in the Earth soil system, where, for example, you can see predicted levels of alpha proteobacteria, which like it dry, in the Arctic and in the Sahara and other dry regions around the Earth. And then what we can do is predict the abundance of different genes using PyCrust, which we introduced to Nature Methods in 2013, and then relate that to predictions of metabolic turnover. And so what's great about this is we can do it under different climate scenarios. And if you think about something like the built environment, where you're still limited by the number of samples you can collect and the number it's technically feasible to process with the low biomass, being able to extrapolate this stuff from things that you can either measure or model within the buildings, like airflow and light and occupancy and so on, would tremendously open up our ability to not just measure the microbiome, but predict what's going to happen under different scenarios and affect it in ways that benefit human health. And so to do this sort of thing, we founded the Center for Microbiome Innovation at UC San Diego a year ago, which basically brings together people in all kinds of different disciplines, from medicine to computer science and engineering, and even to ecology and oceanography, to really address some of these difficult problems. So with that, I'd like to thank the large number of people involved in this research and our various funding sources, many of whom I've mentioned already, so I won't read this out. The many members of my lab, both current and present, who it's been a true privilege to work with. And finally, I'd be delighted to answer any questions you have. Thanks again for the opportunity to do this.